Hello, my name is Mateusz Chiliński and I'm a member of Functional and Structural Genomics at Center of New Technologies at University of Warsaw, Poland. Today I'll be talking about ConsensuSV, Consensus Structural Variant Caller for Next Generation Sequencing for Selected Families from 1000 Genomes Project. What we did in our research, we created a tool called ConsensuSV, and you can see its pipeline on this slide. As you can see, the input to our tool is in the form of VCF files, VCF outputs actually, from various colors. However, it's not limited to those colors because it's really easy to simply retrain our software to take fewer or more colors. You can also use our pre-trained model to establish consensus based on a subset of those tools. We simply take those VCF files, we pre-process them, and then we use decision trees and neural network model to establish the consensus. We also use a voting algorithm for establishment of the genotype. After creating ConsensuSV, we also created functional analysis tool, and you can see its pipeline there. As you can see, there are 5 inputs, however some of them are optional. The first input is structural variants, for example from ConsensuSV. Then SNPs and indels, we use the ones from BCF tools, and we didn't need actually to get consensus there because we mostly operate on the short read data, and it's sufficient to just use one tool for SNPs and indels. You can also provide healthy variants. It's optional parameter. However, it's really useful in terms of filtering healthy common variants or the variants that are not really interesting to us in terms of the analysis. Also, you can put as an input anchor positions as a result of ChiaPet experiments. We also provide the genes annotation file, which is pretty much default, and it's provided for the newest reference genome. After preprocessing the file, as you can see, we go to 2 ways. The first one is the simple 1D analysis, and we look for overlapping sites between SVs, SNPs, indels, depending what's provided there, and the areas of interest. By areas of interest, we mean exons, introns, and also promoters, which we consider to be up to 1000 base pairs from the start of the open reading frame. In case of the ChiaPet experiment results are provided to the tool, we also do the 2D analysis. As you can see, we are looking for overlapping sites between SVs and anchors, and in case it's overlapping, we simply consider the whole CCD. We simply look for all the areas of interest in the CCD where the anchor is compromised, and we put it on the final list. After we establish the list, we use ensemble and UniProt AP to find the actual list of the genes and biological pathways that might be altered. The output from this tool is in form of the CSV file, so it's really flexible for the researcher to decide what to do with that data. Some results which we obtained using our tools, as you can see, we run the ConsensuSV on samples from 1000 Genome Project, and we obtained those are the SVs that were obtained after running them through our tool. And then we actually used a functional analyzer for the filtering. As you can see, we used some pretty basic filtration criteria, and we provided the files from GNOMAD 3.0 with mineral frequency above 1%. We use dbSNP, we use dbVer and CleanVer as well for the filtration of the results. The results of the ConsensuSV actually are right in this table. We compared ourselves to a gold set that you can find in a great publication reference there. And as you can see, we actually compared only Illumina data, not the final output of that research. We also took a subset of the results from Illumina that also covered the tools that we actually use in our research, and we filtered out those that were not reported in the study by fewer than 3 tools, because that's the criteria that we also used in the ConsensuSV getting of the consensus. And as you can see, we got on average 85%, which is, well, quite nice, and we are really happy about those results. I bolded some samples there because we used those 6 samples for training the model, so those were, well, kinda like verification set, and as you can see, it works pretty well. The output of functional analyzer is in form, as I said, of CSV file, so it's up to researcher how actually he or she wants to process it further. However, it is an example of how you can get some data out of it. As you can see, we generated the statistics of areas of interest that were in the CCDs affected by SVs. Of course, we still need to do some filtering, especially in the CCDs that anchors are compromised by the SVs and SNPs. However, as mentioned on this slide, we'll probably go for a QTLS analysis, and we're hoping to get some results really soon and publish the paper. Thank you for your attention. If you have any questions, please let me know.